Thanks very much for the introduction, Dr. Mundus. And the one thing you left out in that lab, we have a really great group of people as well who are producing all this research. So I get the opportunity to speak about some of it, but know that there's a great team at C-Spine who's really responsible for cranking out the work. So I'm going to focus a little bit on biologics and a bit more of a basic science presentation about which cells really work in spinal fusion. So every time the biologics person gets up and speaks, we always talk about the three O's: osteoconduction, osteoinduction, osteogenesis, the scaffold, the signals, and the cells, and iliac crest and the patient's own bone is always, in the middle with all three. But first question we're going to ask today is, does autograft really work? And then if yes, do we really know why? Really basic questions. You'd think we know the answers to these already, but there is a paucity of data. So iliac crest, and specifically in posterolateral fusion, there are a plethora of clinical studies showing the expected fusion rates. And down the bottom, you can see that if you sum them all together, you have an approximately 74% fusion rate for iliac crest in the posterolateral fusion space, ranging from 65% all the way up to 92%. This includes both instrumented and non-instrumented cases, but if you pass them out, you see a significantly higher fusion rate with iliac crest at about 82% when instrumented, compared to about 52% when not. So why don't we use iliac crest on every case then, if it seems to work so well? Well, there may be morbidity from taking an iliac crest harvest, and some authors have found 30 to 40% chronic pain associated with that crest harvest. Others say maybe that crest harvest is overstated. Maybe it's not as bad as we think. This was an interesting study I was involved in the planning of, where they took a unilateral iliac crest harvest and then postoperatively asked the patients to point out which side of their crest the iliac crest harvest was taken from, where's the pain coming from, and quite often, more than not, they could not actually tell which side their graft was harvested from. This is in the context of lumbar deformity surgery, but an interesting finding nonetheless. So often thought of as free, but it's not really free. In the sports study, they actually showed that the cost associated with patients who were treated with iliac crest versus BMP2 showed that there was actually no difference when you summed everything up in terms of the costs therewith. So is it really the gold standard? We hear this a lot now. Yes, it may be, or it has the most data, but is it really performing better than everything else? This is actually the sports study I referenced before, where they showed fusion rates using iliac crest and without, and showed pretty much the same fusion rates no matter what you used. So you look at the outcomes of autograft coming from various different sources. So the first one here, local bone versus iliac crest. This one was actually in PLIF, 101 patients, and actually found equivalent fusion rates, whether the bone came from a local source or from iliac crest. Another study on the right here, in this case, iliac crest bone graft in posterolateral fusion, showing 75% fusion rate for crest versus 65% for local bone. What they actually found in this study was for single level posterolateral fusion, they had equivalent fusion rates between crest and local. But once you got up into a multi-level, then the iliac crest seemed to perform a little better. If we look at where viable cells are, since the focus of the talk today is going to be on the cells from different sources of iliac crest, we can see this paper on the left here, Gertz, showing that the osteogenic potential of local bone was actually equivalent to iliac crest. Another paper over here showing that the osteogenic potential of cells coming from the vertebral body are actually superior than those from the iliac crest. We often hear about, well, what about bone dust, bone shavings that are collected in a trap? Is there anything really alive in that compared to bone that you're taking either locally or from the iliac crest? And there are a handful of papers out there now that that show, yes, there are actually still some osteogenic components retained in bone dust and bone shavings as well, interestingly. However, multiple papers here on the right showing that the osteogenic potential from that bone dust is a lot lower than bone chips. So while you can retain some osteogenic potential from bone shavings and bone dust, it's not quite as robust and viable as you would expect from morselized local bone, as you would expect. So let's get into a little bit of science. And our question here is, if we talk about our iliac crest with all the components and including the cells, well, what really is the contribution of the cellular fraction for promoting fusion? Or in other words, do the cells even matter? So the experiment that we ran here is we took a rabbit spinal fusion model, very well validated with iliac crest autograft at 8 weeks, initially described by Bowden, and now required by the FDA for any new biologic product that we'll try to bring onto the market. The study design here, and Dr. Mundus is actually involved in the planning and publication of this as well. We just recently had it accepted in part to SRS. But the design of this study, our control is a standard viable iliac crest autograft. Our first test group is a partially devitalized iliac crest. That's where we just rinse off all of the non-adherent, non-adherent cell population. So the BMA essentially is rinsed off. And then the fully devitalized iliac crest, we rinse off all of the non-adherent cells and we kill all of the cells that are remaining attached to the bone by a freeze-thaw cycle. On the left, you see a micro CT of what a fused spine looks like, and you can see the clear bridging from transverse process to transverse process. And here in the sagittal plane, we see a really nice bridging here of bone. So very clearly fused on the left. On the right, you see what a non-fused segment looks like and a clear gap right through in both planes. So let's jump to the results. Our viable iliac crest should get around 60 to 70% fusion in a rabbit, and that's exactly what we found in this study. We had 7 out of 12. Our fully devitalized iliac crest with no cells obtained 0% fusion. That should be a pretty shocking result, actually. If you think about it, we took viable iliac crest, we tested and we obtained fusion, but we kill off all the cells and we get no fusion. That really means, or what it's telling us here is why does iliac crest work? It's not just because it's a good scaffold. There is something about the cellular component that's really contributing to that fusion. And I think this is something that we all knew already, but hadn't been published yet, surprisingly. Now, if we take that partially devitalized iliac crest, so again, this is the case where we take the bone and we just simply rinse it in saline to remove the non-adherent cells. And in this case, our fusion result, surprisingly, was slightly higher than the viable iliac crest group at 12 out of 14. So what did we learn here? We learned that the cells are very important. And as we try to narrow it down to which cells specifically are important, it seems to be the cells that are attached to the bone itself, not those that are free floating and easy to rinse away. There were a couple of animals, if you're looking at the numbers and thinking why they didn't add up, we did have a couple of rabbits that didn't make it and they weren't replaced. So our conclusions, again, the viable crest led to fusion, the devitalized led to no fusion. So do the cells matter? Yes, they do. Now that we know that they're important though, our next question is, well, what happens to all that bone as we're harvesting it in our procedures and leaving it out on the back table where it can essentially dry out with time? So this is actually an intern project from a couple of years ago. I bring that up because our interns just started last week. Our first tranche of interns just started, so we're really happy. In fact, I see one on the phone today. Morning, Ryan. And so our objective in this study was actually to again look at the cell viability as we're taking freshly harvested bone and just letting it dry out on the back table. We did it with sheep bone. We've now repeated this with cow bone as well. We morselize it. We check the cell viability versus time. And jumping straight to the results, this is what we got. You see a very typical decay curve, and it actually starts to decay very quickly. So you look at 1 hour after implantation, the cellular viability has dropped by 50%. And 4 hours, the cell viability has dropped by 30%. We've also shown in other studies that you can arrest this cell degradation simply by keeping the bone moist with saline. So the significance of the study, performance of autograft really does depend on the cells, but the bone cells die off very quickly when they're left out under dry conditions. So the cells in autograft really matter, but they are delicate. What could you do with this? There are some things that can be done intraoperatively. We know that changing the workflow to harvest bone later than sooner is likely not a viable option in most cases. But if you're taking bone and running it through a bone mill, you could run it through the bone mill later rather than sooner. So it's not being exposed to as much air as much. And then secondly, you could wet down a piece of gauze and lay it over the bone with simple saline just to help keep those cells viable. And this is also, a part of this was also one of the best papers at NAS of last year. So now I'll turn my attention to cellular bone matrices, big market based around a viable cell component. A lot of products on the market, some are no longer on the market, but is there really good data to show that these cells actually do anything? We just showed pretty convincingly that the cells in autograft bone really have an effect, but no one had published any data to really look at cellular bone matrices, isolate the effect of the cells, and see if there really is a benefit. This paper was just published in JBJS at the end of 2020, but what we did was take 2 of the market leading cellular bone matrix products, test them with and without the cells to see if there's a difference. The products that we tested were Osteocell Pro and Vivigen Formable. The reason that we selected those 2 was to try to bookend all of the different products on the market. Osteocell Pro supposedly has the largest number of cells of all the marketed products and the highest number of MSCs. Vivigen has the lowest number of cells and supposedly no MSCs. The other difference was the DBM substrate. Osteocell Pro has a small amount of DBM particles, and Vivigen has a larger amount of DBM fibers. We then compared it to one of our products, which is 100% DBM fiber-based product without any cells. So first you have to select a model that's valid, and luckily for us, there's the athymic rat model, which allows us to implant human cells in DBM without rejection. Multiple authors have shown that if you take the right population of cells, a culture expanded human MSCs, for example, and implant them in a rat, you can get bone formation. So it's a really good model for this sort of study. Second, when we take these cell-based products, we confirm the cells are actually alive before we implant them. And what we showed is it lines up exactly with the published results for both products, and that our method of killing the cells was essentially 100% effective as well. The way that we kill the cells is simply lyophilization, which is just freeze drying, and we do that to the DBM product as well. It does not affect the DBM or the osteoinductive potential of the substrate. Third, we have a fusion assessment. We do this by micro CT. We have this 3-point scale for quality of fusion. So not just looking at bridging, but looking at the overall quality and maturity of that fusion. On the left, here we can see the transverse process. And then between the transverse processes, between these lines here, there's no bone formation here. This is a clear non-union. As we go all the way across to a 3, now what we see is complete cortex formation, trabecularization. That's sort of the best fusion. And for this study, we looked at a high quality fusion being a 2 or a 3. So jumping to our results, Osteocell, when the cells were devitalized and viable, obtained 0 out of 18 in both cases. So no difference in fusion results due to the inclusion of the cells. Vivigen formable, devitalized, and viable, 11 out of 18 and 10 out of 18 respectively. So again, no difference with the inclusion of the cells. So 2 of the market leading products, again, trying to represent the whole space of cellular bone matrices, show no difference due to the cells, even though we're in an athymic rat model that will not reject the cell component. Then OsteoStrand Plus, the DBM product, getting close to 100% fusion here at 17 out of 18. And if you're asking yourself, why are there all these differences and how could a DBM alone possibly outperform some of these cell-based products? I'm going to explain that one on this next slide. So our conclusion, the 2 products, there was no difference with or without the cells. A very controlled study where there's only one variable, the presence of the cells. And since there was no difference, that means there was no effect of the cell component. To explain the differences between the materials, to explain why Osteocell Pro was not as effective as Vivigen, which is not as effective as quote unquote just the DBM, you ask yourself, if the cells don't work, then what is left behind to drive the fusion? With Osteocell Pro, it's mostly bone chips, Cancell's bone chips, and a smaller component of DBM particles, which is the less effective type of DBM. Vivigen has bone chips and it has a larger amount of DBM fibers, which is also a better type of DBM. So Vivigen outperforms Osteocell because it has a better type of DBM and more of it. So the conclusion here is that the fusion in cell-based products is not being driven by the cells, it's being driven by the DBM, but they only have a small amount of DBM in them and sometimes a small amount of a not very good DBM. Versus the product, which is the 100% of the active material performing closer to 100% fusion in this model. So the explanation is actually very simple, that the fusion is being driven by the DBM. So perhaps we could just be more cost effective and have better clinical value by simply using the DBM by itself. So I know we all are interested in rat fusion studies, but even more so interested in human data. And I just pulled up some of the Osteocell or cell-based product data in ACDF. So 3 main papers. We can see the one on the left by our own Dr. Islak as the lead author. And a quick summary, the paper on the left in 2014, 182 patients, 1 to 2 level ACDF with a result of about 87% fusion by CT. In the middle, 57 patients, again, 1 and 2 level ACDFs, 87.7% fusion. And a new paper on the right that was just published within the last week or 2, at least the last month, 326 patients, single level ACDF only with 86.8% fusion. And if you put them all together, 563 patients and 87% fusion rate in ACDF, which I think most of us would argue is less than we would expect, particularly for the cost associated with these products. So just to kind of wrap it up here, which cells really work in spinal fusion? Cells in autograft work. We showed viable iliac crest fuses a rabbit, but devitalized iliac crest bone graft does not. The cells attached to the bone seem to work harder than those that are not attached to the bone. Cells in cellular bone matrices seemingly don't work. We could not get them to improve fusion in a rat, suggesting that even though there are living cells, they may just not be the right type of cells. And with that, I wish you a good morning. Any questions?
Thanks, Frank. Awesome. That was awesome.
Welcome.
Hey, this is Hanny here, and I just wonder, your opinion as to why there's still so much investment and work and sort of activity around the cellular bone matrices, despite the evidence that you've shown. Because among surgeons, I think the traditional knowledge is sort of just what you mentioned, that there's not really sufficient evidence to justify the cost of the cellular bone matrices versus traditional DBM. I thought that the results are equivocal. I didn't realize that they were actually inferior in some instances as well. So any insight into why there's still so much emphasis on that, at least from an industry side?
Well, yeah, interesting. So I'll answer the scientific side first, and then I can answer the industry side. On the science side, I think the initial concept made a ton of sense. I mean, when they were initially launched back in the day, it was kind of branded as tissue engineering and stem cell products. But in reality, what made more sense to me early on in the development of these products was that it was just like iliac crest in a jar, right? It was like autograph that you could take out of the freezer and implant into anybody. It had all three components, the three O's, right? Osteoconductive, inductive, osteogenic. So it just seemed like it made sense. And where the science got to was really well characterized, that those cells are truly viable after you cryopreserve them and then thaw them out. They truly are viable. You can get them to differentiate into different types of cells. But the science ended with what happens when you implant them. So, I think it generally sort of made sense, but where the gap is what happens to these cells when they're autologous versus allogeneic. You take your own cells and you implant them back into you, they'll survive, they'll persist, they'll do their thing. If you take my cells and implant them into you, my cells will likely die off very quickly. And they call it this trophic effect, right? Where they release all the growth factors that they have. And that story just doesn't resonate as well as the multi-lineage, differentiation potential of MSCs. But on the industry side, there is always interest in selling a product at a higher price point, frankly.
Yeah.
Right? And that's what these are. So that's what sort of drives a lot of this. And most major companies now, I think maybe C-Spine is the only exception. Most spine companies carry a cell-based product. Although I suppose Medtronic has an asterisk next to it based on their recall that we heard about a week ago.
Yeah. All right. Thanks, Frank.
Hey, Frank, the original allograft cellular matrix, as you said, was mainly basic science driven. But I believe there was wasn't there some rabbit model basis for considering it, which just wasn't the same rigor from an animal model as the athymic rat in the kind of thoughtful methodology you utilize to really compare against other types of products and ferret out what the merit was on a basic science level.
Yeah, others have shown, there's quite a few other papers now where people have taken stem cell based products and put them into, say, a rat. But what they've done in that case is just take the product off the shelf and test it. What we did that was different was the with and without the cells component. And then during the development, mostly what they were showing was actually cultured MSCs attached to these products. So they might take, say, a sheep, and you're going to do a fusion in a sheep. So you would take the sheep MSCs, culture them up, you'd add them back onto the substrate. So it's kind of different than what these products ended up being. But to get them on the market, because it's a tissue-based product, you really just have to prove safety. You didn't have to prove efficacy. And then once there was one on the market, then there became two, and now there's 20 or so.
It's a very good learning experience, I think, for everybody to be a little bit skeptical when new products come on the market. Be thoughtful about the basic science foundation of them.
And you'd be shocked actually with some of the basic science, like even this autograph stuff we're doing. You think that data is already out there, that people have kind of nailed down that yes, the cells are important in iliac crest and which type of cells, but there really isn't that much scientific evidence, really. It's kind of, we all just assume it. Yeah.
Oh, very good.
Awesome,
Thank you
Frank.
very
Really
much.
appreciate you. Anybody else have any questions?
I had a comment. I really enjoyed your talk, Frank, and there's no question that biologics is going to be, the way of the future. And the more we understand how they work, I think we can use it better. my comment just was, being an old timer, was about iliac crest. And I think the studies that have been out there, it's very hard to compare them together because it's hard to really know how they have done posterolateral fusion, for instance. Is it a transverse process, overlay bone graft? It is inlay bone graft. Are there been any facet fusion where the cartilage is gone and you put bone graft between the facets? None of them describe the detail. Not really comparing the apples to apples. So you have to be careful about the mixing all of these together as far as the iliac bone graft is concerned. But there's no question there is not much time and effort now to do a proper posterior iliac and facet fusion. So I think your effort and in general biologic should move forward, and hopefully we get a means of effective and cost effective and time effective way of fusing spine. Hopefully we don't have that for a long time because we like to keep the mobility of the spine, not the fusing the spine. Hopefully that's for many years from now. Thank you for really good talk. Thank you.
Well, thank you as well.
Well, I don't think it could have been said better. Thanks, Dr. A. And if anyone else has any questions, go for it. Otherwise, we're going to let Frank get back to doing his thing and
I have a real quick question. May I ask a quick question?
Of course.
What are your thoughts on iliac crest bone aspiration with like a Jamshidi needle?
Yeah. So certainly not against it. I like the idea of the patient's own cells. What our data just showed, essentially where we tried to separate the cellular component into two. So we took the non-adherent cells. So those would be the cells that you could aspirate off. And then we looked at the cells that were attached to the bone. And it seemed that it was the cells that were attached to the bone that had the greater effect in fusion, at least in our study. Now, this is just one, rabbit study, so it's obviously not the rule. But we seem to suggest that the cell population you could aspirate off was not the cells that were responsible for the fusion.
So basically, you'd be better off taking a core out of the crest than to do a Jamshidi aspiration, right? So if you're going to put a little hole there, you might as well make it just a little bit bigger and get a core biopsy out of it, so to speak, and that way you get some bone with it.
Thank you.
Awesome. Well, thanks, guys. Thanks, Frank.
Frank.
You're welcome. Thank you all.
Everyone have a wonderful week.